For our team project, we decided to focus on human-induced pluripotent stem cells and their application in discovering therapeutics to treat ALS patients. To begin the process of inducing pluripotent stem cells, somatic cells are taken from a patient, and through the introduction of identified transcription factors that promote stem cell identity, such as KLF4 or SOX2, the somatic cells go back to a pluripotent state. The cells can then be reprogrammed to differentiate to any relevant specialized cell. The advantages of this approach is that it avoids ethical concern and controversy involved with research using embryonic stem cells and provides scientists with a more relevant human model for research. HiPSCs can be used to study complex human diseases, specifically neurological disorders such as amyotrophic lateral sclerosis, or ALS. Studies of the central nervous system have been difficult to investigate because it is hard to obtain live brain tissue. However, the implementation of HiPSC technology has allowed us to study human brain physiology and neurodegenerative diseases. HiPSCs are derived with known disease-related mutations. In vitro differentiation then occurs into their respective cell types. This can result in differentiation into any disease-relevant cell type. Cell types within the central nervous system include specific neuronal subtypes like astrocytes, oligodendrocytes, and microglial cells. This provides a model system to study complex human neurological diseases. This is used in conjunction with CRISPR to produce or fix human model systems, and then used to provide insight into the molecular mechanisms of the disease, as well as to observe and develop novel therapeutics. Scientists use this approach to investigate novel therapeutics to treat ALS. In ALS patients, iPSC-derived motor neurons have increased excitability compared to wild-type cells. Scientists targeted Kv7 channels, a voltage-gated potassium channel, for correcting hyperexcitability because they are activated at subthreshold voltages, which makes them regulators of excitability. So, retigabine was already a clinically approved anticonvulsant and is a specific activator of subthreshold Kv7 currents. So they hypothesized that it would correct the mutant. They found that retigabine increases in vitro survival of ALS motor neurons, confirming that motor neuron hyperexcitability contributes to motor neuron degeneration in ALS. HiPSCs have shown to be effective at reversing disease-associated phenotypes in vitro, however, only in the early age of onset disorders of neurodegenerative diseases. Most of these diseases do not show symptoms until it is already too late to be treated. There is also uncertainty of whether cell culture models for late age of onset neurological disorders could identify any disease-relevant phenotype. There are current efforts that attempt to mimic age-related processes in cell culture using a gene called lamin A that is responsible for premature aging. Clinical trials have begun evaluating the effectiveness of HiPSC-derived therapeutics for transplantation and novel drugs developed in HiPSC disease models. This is important because candidate therapeutics that are discovered in vitro might not have the same effect in vivo. In hopes of further technological advancements, we believe human-induced pluripotent stem cells show great promise in the future of translational medicine.